A very good afternoon to all of you, my name is Dr. Priya Agnihotri, and all the work that I'll be showing today has been done under the funding of Birbal Sahni Institute of Paleosciences, Lucknow, and today I'll take you to the world of fossils, where I would like to showcase a few collection of arthropods that has been found in Indian amber. That is a plant resin. I'm going to explain it later. From the Cambay and the Kutch lignites of Gujarat, and what are the paleo-environment implications. Fossil arthropods, they are significant because they have been megadiverse for some 520 million years, and they became abundant in the Paleozoic, but however, they radiated during the Cenozoic time period, that is starts from 66 million years ago. And studying them afford glimpses of the vanished morphologies, sometimes extinct in the modern counterpart, and they reveal patterns in ecology, phylogeny, and occurrence at the time of splitting events, aid in tracing lineages, evolution, and geographical distribution which are subject to migration and dispersal. Amber is an excellent source to preserve such fossil elements which are significant in geological point of aspect and also in building up fossil forest. We can say an ancient forest where different kind of organisms are there along with the flora and the fauna and other elements in the ecosystem. This is a representation of what amber has formed, how it is formed. That is actually resin that flows down or in different ways from a tree bark, and it gets fossilized above 1 million years of age, and common in the lagoonal deltaic environments, produced under definitely abiotic stress or as a defense mechanism of plants against insects, and they serve as an exceptional three-dimensional preservation which is significant for taxonomy and fossil identification. And definitely with these collection we can form and catalog fossil biodiversity and study different association of species. This is the view of a lignite mine at Umarsar, Kutch Basin. These black strands are the sediments where we climb and we go and we search for lignite bands, and there we hunt and dig for amber nodules which we bring to the laboratory and then refine to find the insect or any fossil biota that we have. And these have been potential resources for paleontological evidences which are pertaining to palynology, vertebrate fauna, megaflora, and significant hotspots definitely for amber collection. Now, this is just a pictorial representation to show you that amber has been studied across the world. They have a different physical property, different chemical property, different botanical resources, and in this regard, our Indian amber is definitely unique, because the plant source that exudes this resin has not been found anywhere apart from India, and that belongs to the Shorea trees, Angiosperm family, and Dipterocarpaceae. So that is not found anywhere in the world. That's why studying Indian amber is unique. Coming to the biodiversity in amber, in Indian amber, so we have the maximum of flying biota. We have categorized into canopy biota, which holds 55%, and next followed the terrestrial or the bark dwelling biota that includes social insects, the ants, the bees and wasps, and also arachnids. Then we have the freshwater biota, that is aquatic fauna. We will discuss it because ostracods are very important when we find them preserved in amber, and least amount is the subsoil biota that includes beetles and mites. Now, what is the significance of arthropod fossils is during the time around 88 million years of age, the Indian plate suffered a lot of tectonic, or you can say thermal experiences, thermal events which caused radiation and different forms of adjustments for the biota that lived there, and one of the category was insects as well. So that's why studying those in that time period, it gives you a data of coevolution, forest ecosystem, and we build up, we kind of reconstruct what kind of a forest existed during those time when Earth was in Eocene and a tropical Paleoposition state. So this is some of the Eocene arthropods in amber. On the left is the Chironomus midge that has been preserved with its delicate antenna and eyes. And then we have the Formica, the Dolichoderus ants that have the dominance in the social events, followed by some wasps as well. So since I think today is the spider day, so we have some spiders as well. And this just below that you have the Geogarania valiensis. This is the Indian representative of the pseudoscorpion, just published a few months back, and it is the only phoretic insect found in Indian amber, and not recorded anywhere from the other literatures. And this is the Sarcoptes mite. We have done a scanning electron microscopy. We have dissolved the specimen, we have extracted the mite, and then we have imaged it under the scanning electron microscope. So there is a lot of possibility when it comes treating or playing with amber rocks. See, I told you, a day of spiders. So we have the evidence of fantastic depiction of spider webs that suggests that the silk threads acted as a protective covering for the egg-sac spiderlings and to capture prey that included roaches and millipedes. Hence, their existence can also be manipulated in that way, and they trap both on ground or on tree branches for trapping hordes of flying insects. The egg-sac is definitely under suspect because we went through CT scanning. We found some high density molecules inside this sac, so we think that this egg sac may contain some spiderlings as well, but we are still susceptible about it. Next is the least but significant Phytophagous and thermophile group, includes the beetle, and this is a CT scan picture, and the red area is the more denser part. Next is this Auribatid mite, S. cachensis. This is also a new species, has been Indian species, and this is a perfect example of how coevolution can exist, and how an insect can evolve from one particular thing to another feeding mechanism. So probability of mite families, they adapted to parasitism as mammals also co-evolved and diversified during this time period. So this is just to show that Diptera, that are the flying insects, the canopy biota, had the highest and the dominant insect assemblage that was there in the amber collection up to date. Okay, now we talk about insects, plants, trees, that's okay. Okay, one can think that okay, the resin is falling and one is going to attract whatever it takes with it, like on trees, insects, everything. But amber preserving aquatic fauna is a point of question. So the hypothesis is that around the area where insects or the resinous plant was there, it was surrounded by a water body. So these resinous trees had hanging branches. So when the resin actually fell down, it reached into the water body and captured these delicate aquatic crustaceans. Pictorial representation is that of a unique ostracod trimiciviera, and it has a gigantic size of 5 mm, and along with these, it also holds very well-preserved soft parts like the appendages, antennules, which are definitely very difficult to find in sedimentary fossils, and activity can also be found like the open brood pouch. This ostracod is bursting out eggs, so we capture life forms in amber, so that's striking and fantastic to study further. Along with this, definitely other elements would have been there. So we have studied the biotic elements, coexistence of pollinating insects, the flowers, and this is how we reconstruct a kind of angiosperm forest, and that includes cohesive groups. Also, a very fantastic thing is the kind of volcanic sources we can estimate from elements found, and also a lot of floral and faunal connections, faunal connections. So this is great in like deciphering about fossil forests, and that existed during the Eocene era that leads from 55 million years to around 40 million years. What happened into the forest? In a nutshell, it provides details about the past distribution of plants and animals that are used to reconstruct environments in different regions of the world, and definitely a source of micro and macro evolution, and it definitely serves to study the paleontological history of biological diversity and association with other species in a particular habitat. Thank you for your patient listening, and this is the field work that we did. That's my supervisor, Dr. Hukam Singh, and we had made a collection of amber fossils. So thank you so much. Looking forward for some questions.
Thank you so much. It was a wonderful talk. Do you have any questions? No questions? Yeah. Oh.
Yes, Kunte sir.
Hi, nice talk. I'm always fascinated by the insect communities that you find in these amber deposits. So if you look at the diversity that you see in amber, there must be species which are more common than other species or groups. If you look at modern day insect faunas or invertebrate faunas and look at which groups are abundant, do they correspond with these amber collections? In other words, have some groups of insects in
like the modern counterparts.
Right. So have the groups of invertebrates you find in amber, have those changed over time? The composition is
in fossil concept, or when you're comparing it with the modern one?
Both.
Like with both. If we take two different localities and then we study arthropod or insect collection from one area as compared to next, there is definitely differences. For example, if we take example of Baltic, suppose Baltic or Rovno amber, they have pseudoscorpions in immense number, but in Indian amber we have just recorded one till date. So definitely, if you compare with geographical locations, different geographical locations, there are different kind of collection you will see.
Well, let me ask this differently. Maybe that will make it clear. Do you see any evolution of communities or groups of invertebrates when you compare fossil record through time? These amber deposits.
Like the course of evolution, how that particular thing has evolved with time. Right.
Sort of like that. So earlier, for example, there were lots of pines and those kinds of plant species. Now you see many more angiosperms. That kind of faunal change I'm talking about in the fossil record.
We studied about, with this respect, we studied using the traits, the characters. So few characters were not in the present forms, but they were present in the fossils. So this is how things have changed, and insects have co-evolved. It has co-evolved.
I'll talk to you more about that.
Yes, definitely. Thank you.
Thank you very much.
Thank you.
This is excellent work. I have few queries. How you decide this amber is from Soria or from some other plants?
We have done biochemistry on the kind of resin. Like there is a resin, insect is trapped. We take that resin and we have done with biochemistry. We found the elements, and those elements were similar to that particular kind of family and of plant. So that's how we estimated.
So what are the particular techniques?
Techniques like
I mean this biochemistry, which kind of techniques you are using? I mean
we have GSMS and every other thing, like there's a team doing that. We have just got the data, and there's a number of other things as well, but we have petrology and other palynology things, and we go through different kind of instrumentation techniques.
And in our Indian scenario, there is resins from Boswellia. There is another category.
Yes, sir.
And also, I don't know from which plant it comes, something we call gugul, that we use during different puja.
Yes, sir.
Have you thought about those kind of ambers, whether they are got fossilized or?
See, there are differences in amber, copal, and gum.
Yeah.
So there are different types of things which we confirm using biochemistry. So other aspect is if that copal or gum or whatever is there, if that has a good preservation tendency, or if the insects are there or not. So it depends a lot on the probability and other factors as well. Thank you.